So, welcome to Bio 212, Anatomy and Physiology 2, Norwalk Community College. And we are about to finish up Chapter 19. And what I want to do is just sort of remind people what we've covered so far, where we actually have material inside a closed circulatory system when we call it blood. And that blood is broken up into parts, plasma, which actually has materials dissolved inside of it. You actually have a buffy coat, which are the white blood cells and the platelets, and then you have red blood cells. And so far we've broken down what each of these is doing, right? So imagine, if you will, what we've discussed are, hey, what are those white blood cells? And how are they, generally speaking, being activated, and what are they doing? Likewise, red blood cells, we told you they carry oxygen, carbon dioxide, and nitric oxide, and they have a specific lifetime. They require a specific hormone for them to be made. The activation of that, it requires hypoxia. And then we just finished up clotting, where essentially the platelets, which were found down here, release materials that interact with soluble proteins that we found inside the plasma of blood, to go from a soluble protein, which is the fibrinogen, to then make fibrin threads. And then inside of those fibrin threads, we put in proteins that would allow for the dissolution of those clots. Okay? What I want to do now is I want to talk about what are the types of red blood cells that you find and where they come from. And this actually is a review. So if here's our red blood cells, we know we have our red blood cell over here, biconcave disc, and it's carrying all sorts of oxygen, carbon dioxide, and nitric oxide inside of it. But remember, on the outside of the cell, we have these glycolipids, and these glycolipids can come in the form of A or B, okay, or AB, or none. Okay, so none of those could be present, right? And that's a significant idea, because that means your body knows what types of red blood cells it has. Okay, for the same reason that up here we have antibodies that are soluble, that were made from those B cells, and they're constantly looking to see, hey, do I have antibodies against A? Do I have antibodies against B? Okay, do I not have any antibodies at all? Okay, and if you can answer that question, then you can understand what we're talking about. Okay? Meaning, if your body can answer that question, that means you can understand as a student what's going on. Okie dokie then. So what are blood groups? Let's go. Where did you go here? Let's just go right here. So here are your blood groups. Here's your basic antigens and antibodies of the ABO blood typing system, right? Where if you're a A-type blood, it means on your red blood cells you have the A antigen, which means inside the plasma of your blood you have anti-B antibodies. If you're a type B person meaning blood type person, you have B antigens, which means inside the plasma of your blood, you have anti-A antibodies. If you're AB, you have neither antibody present. If you're type O, it means you have both antibodies present, right? We can chelate antibodies out of the plasma of your blood, so therefore you can end up with enriched O-type red blood cells, and you can give those to anybody, okay? That's why O negative, Rh negative, right, would be the ideal blood transfer type. Whereas, obviously, AB would be the least ideal transfer, but you're a universal recipient, okay? Because you have no antibodies against anything. Why do we even think about this? Well, let's tie together some ideas we've covered so far inside this class, and we'll finish up this recording in just a couple of minutes. The body itself has an immune system, and that immune system is always looking for other, okay, for reasons well beyond we've been paid to understand at this point in time, or you've paid to understand, and what we'll do is we'll definitely come to inside chapter 22, is the mom's immune system, if mom is Rh negative, is making antibodies if the first fetus was Rh positive. Okay, she will begin to make antibodies against the Rh antigen. So that means as the second pregnancy comes along, those antibodies will have the ability to cross the placenta into the fetal circulatory system and now act against the fetal tissue. Now, this isn't a big deal anymore. I mean, you give somebody Rogam, it binds those antibodies, and next thing you know, you have a completely normal fetus to term. What it does, though, is it does allow us to sort of come up with a mechanism to begin to understand. Hey there, girl. How you doing? Begin to understand how it is the immune system is actually going to work. And that's what I'd like you to walk away with. So we all have a circulatory system, and the fetus has this material. And the material now is passed through the placenta into the body, which means now the body can begin to identify this as other. And that's going to cause the production of specific types of antibodies. And that's where we'll finish before my cat gets over here and starts meowing. Okay, have a great day. Bye.